classification is based on how messenger RNA is produced. Now, group 1 is easy because in group 1, the viruses already have double-stranded DNA. So the double-stranded DNA undergoes transcription normally. It's transcribed into a single-stranded messenger RNA. But then in group 2, the genome of the virus is a single-stranded DNA. So first, the single-stranded DNA is converted into double-stranded DNA using DNA polymerase. This method is similar to DNA replication, except instead of making 2 molecules of double-stranded DNA, you're only making 1. And then after that double-stranded DNA is generated, it can be used directly in transcription to generate a messenger RNA, a single-stranded messenger RNA, using RNA polymerase. So that's group 2. And then group 3 is double-stranded RNA. So the double-stranded RNA simply is used to transcribe a single-stranded messenger RNA. So instead of using DNA polymerase, RNA polymerase is used to convert the double-stranded RNA into a single-stranded messenger RNA. Group 4 and 5 are both single-stranded RNA viruses, but group 4 is a little easier to understand. Single-stranded positive sense viruses, single-stranded RNA positive sense viruses use the single-stranded RNA directly as messenger RNA. So in other words, group 4 viruses skip transcription. They do not even have to go through the process of transcription because their single-stranded RNA genome is already used as messenger RNA. But in group 5, the single-stranded RNA is a negative sense molecule, and so it undergoes transcription and is converted to another single-stranded RNA in the form of messenger RNA. And that process uses RNA polymerase. So group 5 is single-stranded RNA, but that single-stranded RNA is not directly the messenger RNA that is going to be used to make proteins. And then we have group 6 and 7. Group 6 is single-stranded RNA viruses with reverse transcriptase. So what happens there is that the single-stranded RNA is converted to double-stranded DNA, and this process uses reverse transcriptase because it's the reverse of transcription. Remember, transcription is converting DNA to RNA, so the reverse would be converting RNA to DNA. But once you have your double-stranded DNA, then transcription proceeds as normal using the enzyme RNA polymerase. And then group 7 is a double-stranded DNA virus, but it contains the enzyme reverse transcriptase, and double-stranded DNA is not converted directly into messenger RNA. So first what happens is the double-stranded DNA is converted into an RNA intermediate, and then the RNA intermediate undergoes reverse transcription and is converted into single-stranded DNA, and that process uses the reverse transcriptase. And then the single-stranded DNA is converted into messenger RNA using RNA polymerase. Now, we have talked about classifying viruses because classification is important in understanding how the viruses work and just being able, just like it's important in classifying living things, it's being able to group certain living things based on shared characteristics or shared genetics, shared DNA. So now let's talk about what happens when you're infected with a virus. So what happens, what is the impact of viral infection? The viral replication cycle can produce dramatic biochemical and structural changes in the host cell, which may cause cell damage. May. And if it does cause cell damage, we call that cytopathic effects. Cytopathic effects are the effects. Cyto means cell, pathic means pathogen. So in this case, the virus is the pathogen. So it's just pathogenic effects on a cell. So in other words, cell damage caused by some type of pathogen. In this case, it is a virus. Um, the symptoms of viral diseases result from the immune response to the virus. And the immune response attempts to control and eliminate the virus from the body and also to eliminate or control cell damage caused by the virus itself and the cell that's infected with the virus. Some infected cells, like the common cold virus, common cold can be caused by different viruses. The coronavirus is another one, but in this case, we're going to talk about the rhinovirus because that's probably the most well known, and the most often the common cold is caused by the rhinovirus. So a common cold virus causes the host cell to die through lysis or bursting, or apoptosis, which is basically programmed cell death. In other words, there is a gene within the cell that is designed to activate cell suicide when it is infected with a virus. So the common cold will cause cells of your nasal cavity and your lining your respiratory tract to actually die. And that leads to a lot of the symptoms of the common cold, which are, as you know, runny nose and coughing and sore throat and all of those things. So what happens when that host cell lyses or bursts or else dies by apoptosis is that it releases all its virions, all the progeny virions at one time. And so now all of those virions are free to infect new cells. So many animal viruses, such as HIV, which remember is a retrovirus, leave infected cells by a process known as budding. So in that case, we learned about budding when, I don't know if we've even talked about budding that much, but when we study fungi, the yeast cell, we're going to learn that, okay, here's a yeast cell, an individual yeast cell. And what happens is that yeast cell will form a little bud off the side, and eventually that little bud will become its own little tiny yeast cell. Well, this is kind of what happens with HIV, except that inside of that X stands for an HIV virion. So in that case, each virion leaves the cell individually by budding from the plasma membrane of the cell. So the cell is not immediately killed, but the cell is damaged, so the cell cannot function normally. And in the case of HIV, it's infecting cells of your immune system. So when those cells don't function normally, your immune system is not functioning normally. And that's the reason why um, when a person starts exhibiting symptoms of active AIDS, that their immune system pretty much shuts down. All right, this is a picture that's showing you the steps of a bacteriophage viral infection. Remember that a bacteriophage is a virus that infects a bacterium. So what we're looking at, we know is a bacterium because it has a single circular chromosome. That purple chromosome is a single circular chromosome. Unlike humans have 46 individual chromosomes, but a bacterium has a single circular chromosome. So a bacteriophage um, attaches, that's the first step, it attaches to its host cell using a viral receptor. And then the DNA from the bacteriophage, we call that phage DNA, is injected into the host cell, and then it incorporates itself. Well, in this case, it doesn't. I'm sorry, that'll come later. So it's injected into the host cell, and then you can see that it replicates. And as it replicates, it contains genes for making the components of the capsid, the outer, the outer portion of the virus. In this case, has a complex shape. It's got a head and then it's got a tail with tail fibers. Well, the phage DNA replicates, and then the phage DNA is used to create the components of the capsid, and then that's the replication part. And then the assembly part is when the phage DNA is incorporated into the capsid. And now you have lots and lots of bacteriophage inside of your E. coli. And then the cell will burst or lyse and release those new virions. So the steps are attachment, entry of the phage DNA in this case, replication, assembly, and release. Now, that is the lytic cycle, but bacteria and other types of cells can also undergo a cycle called the lysogenic cycle, which is this occurs when you have viruses that will go dormant or latent for a certain time. Think of the genital herpes, for example. People who are infected with the genital herpes virus can have outbreaks. They don't have herpes all the time, but they can have outbreaks. Specifically, particularly if they have some kind of stressful situation in their life. Um, it can be just general stress, or it can be the stress of being sick that can trigger the lysogenic cycle to convert back to the lytic cycle. But okay, let's talk about the lysogenic cycle first. As you can see, there's an attachment phase, and then the phage DNA, which is green, is injected into the host cell. But now the phage DNA, which is green, is incorporated into the bacterial chromosome or the host cell chromosome. And every time the host cell divides, so in this case, this would be binary fission, and produces new offspring, the new offspring contain the gene for the bacteriophage. So if a situation becomes stressful, in humans, a stressful situation can be actual stress, um, like COVID-19 has caused all of us, or it can be stress due to sickness, or in a bacterium, stress can be, let's say, the conditions in the environment become stressful. You could have not enough food, not enough water, not enough shelter or space. Maybe the bacteria have multiplied so much that there's not enough space for individual bacteria to get what they need. But the point is, some type of stress will cause the lytic cycle to begin. So instead of being latent, the virus is now active. And what happens there is that the phage DNA removes itself from the host DNA, and then the lytic cycle continues. The phage DNA is used to generate more phage DNA and capsids, and then you have the assembly stage where the DNA is incorporated into the capsid, and then you have the cell lyses and the bacteriophage, virions are released into the environment.